So now, in order to understand the process of translation, that is protein synthesis, first structure of tRNA and function of it or role of tRNA in translation must be understood, as well as role of other RNA. We know what is the full form of tRNA. Children, that is transfer ribonucleic acid. Now coming to part of tRNA involved in translation or protein synthesis. The first part is the anticodon arm. It is also named as anticodon loop. Second is the amino acyl arm. Third one is the T loop, and fourth one is the D loop. And fifth is the variable loop. Now, how they are involved in translation? First, let me show. So this arm is the amino acid attachment arm. This is a specific amino acid attachment site. And this one is the anticodon arm, anticodon loop. And this one is the T loop. It helps in the bind to ribosome. And this is D loop. This loop is known as D loop. It helps in binding of amino acyl synthetase. And this one is the variable loop. It is variable in both nucleotide composition and in length. Now, what is the function? From only name we can understand, like anticodon arm. Anticodon arm. It has a specific anticodon, or what to say, complementary basis to the codon of mRNA. तो यहाँ पे आप देख रहे हो कि specific anticodon present होते हैं against the mRNA, right? So anticodon arm tRNA contain the bases which are complementary, that is opposite to the bases of the mRNA. That is called codon. तो mRNA की जो सीक्वेंसेस होती हैं वो कहलाती है कोडोन, और कोडोन के अपोजिट एंटीकोडोन, राइट? So basis which is present on the mRNA is known as codon, and basis which is present on the tRNA it is known as anticodon. Okay, so tRNA can bind with mRNA due to the complementarity. Now coming to the next part, that is amino acid attachment arm. Actually, this must be discussed prior to anticodon arm. As we can say that at 3' OH end, one amino acid, that is 3 bases or nucleotide, comes and join. I repeat that at amino acyl arm, 3' OH end of tRNA, at amino acyl site, one codon or nucleotide, we can say ribonucleotide, come and join. Later during translation, it is added to the growing amino acid of the polypeptide chain. I repeat, at amino acyl attachment site, that is 3' OHN, one codon of 3 bases come and join. Later this codon or this amino acid, we can say amino acid, one codon for one amino acid, that we studied in the genetic code also. That was it. What it does, this amino acid attach itself with the growing polypeptide chain. तो इस तरीके से यहाँ amino acid आते रहते हैं और जुड़ते रहते हैं और एक polypeptide chain form करते हैं, right, during the protein synthesis. अच्छा, now talking about the function of D loop that helps in the organization. Talking about the T loop, that is T arm. So children, T loop is the specialized region on the tRNA molecule which basically act as the spatial recognition site for the ribosome to form the RNA ribosome complex during protein synthesis, or we can say the translation. ओके, सो जो टी लूप होता है टी आर एन ए मॉलिक्यूल पर, वो एक रिकॉग्नाइजेशन साइट की तरह वर्क करता है जो basically that recognize the ribosome to form the tRNA ribosome complex, right? So this is about the structure of tRNA. Now the next part that we discuss, what is initiator tRNA. The next important thing which I want to share with you all, that what is exactly the initiator tRNA. It is a specific or special type of tRNA which bind to the specifically codon AUG, which code for methionine, right? Then after binding it, then this tRNA starts or initiate the process of protein synthesis. So till now we studied the parts of tRNA involved in the process of translation and functions of each part. So what is tRNA? tRNA is the initiator tRNA. Now what is the secondary structure of tRNA? How does it look like? So initially it is the linear structure, then it forms a structure like this, and this is the structure, secondary structure of tRNA. So the folded structure of tRNA, due to the formation of hydrogen bond at the specific location, it is called as secondary structure of tRNA. Now coming to how does it look like. So children, it looks like a glove. So one more point, who discovered tRNA? Who proposed this model? So children, it was Francis Crick who discovered this structure of tRNA. Now coming to the function of tRNA. tRNA act as the adapter molecule. Now what is adapter molecule? Adapter molecule means that it with tRNA amino acids come and attach it amino acid attachment site. Later the amino acid joined with growing polypeptide chain during the translation or the protein synthesis. That is the main function. So one by one tRNA goes attached with the mRNA during the translation, and one by one amino acids are added to the growing polypeptide chain. So this is the main function of tRNA. In the very simple word, tRNA act as the carrier of amino acids during the protein synthesis. When we discuss about the translation, this can be understood better. Now, how tRNA actually looks like? It actually looks like the inverted L structure, right? Now coming to the translation, let us discuss more about the translation. Translation is the third step of central dogma. It is the continuation after the transcription, as we can say. And during this step, amino acids are added one by one to form the polypeptide chain. And another term for translation is the protein synthesis. So it is the third part of the central dogma in which polypeptide chain is synthesized in ribosomes using mRNA as the template strand and tRNA as the carrier of amino acids. Okay, so from this only we can say the role of mRNA and tRNA. So first one, role of mRNA, as we can say, it acts as the template strand. What does it mean? According to the bases present on the mRNA, tRNA come carrying with the specific complementary amino acids or the anticodon bind with mRNA. So with the very simple words, the bases present on the mRNA, according to that specific type of tRNA which carry complementary bases to the basis of mRNA, come and bind by the forming of hydrogen bond, right? Now role of rRNA, if we talk about, R stands for ribosome. So specifically the small unit, the small subunit, it basically acts as the site for the translation. It provides the site for the protein synthesis, right? Actually children, we all know that ribosome consists of two subunits, one large and one small. So during protein synthesis, this inactive ribosome has two subunits, large and small. When mRNA come and bind with the small subunit, it get activated and then it starts protein synthesis. So in that way it provides the site for the translation, right? Now let us discuss about the steps of translation. Now steps. Prior to translation, what happens before the actual stages of translation? So the very first step is the charging of tRNA, that is amino acylation. अब ये चार्जिंग ऑफ tRNA यानी कि एमाइनो एसाइलेशन क्या होता है? जो सबसे पहला स्टेप होता है वो होता है चार्जिंग ऑफ tRNA, जिसे हम एमाइनो एसाइलेशन कहते हैं tRNA का. What is that? Children, during this step, amino acid come and attach at the 3' OH end of the tRNA. अमाइनो एसिड आते हैं और 3' OH पे अटैच हो जाते हैं. This require energy and that energy is provided by ATP. In the very simple words, binding of amino acid, one amino acid at one amino acid acceptor arm at the 3' OH end required energy which is provided by ATP. And the step of the process, that step of attachment of amino acid is known as charging of tRNA, or we can say amino acylation of tRNA. Now next step. Prior to actual stage of translation, what actually happened? In the case of inactive ribosome, there are 2 subunits, right? What happens in the next step? Inactive case में ribosome की 2 units होती हैं, large unit and small unit, right? To the small subunit, mRNA come and bind. अब क्या होगा? This activates the ribosome. And large subunit has 2 attachment sites for amino acid which is carried by tRNA. Now coming to the stage of translation. Translation occur in 3 major steps. What are those 3 major steps? That is initiation, elongation and termination. Now let us discuss about each of these in little more detail. First step, what I told that prior to initiation, this happens with the inactive ribosome with the small subunits. Okay. Now mRNA attach itself, and after it what happened? Amino acylation also done. So what happens during initiation? During initiation, initiator tRNA come and bind with the specific sequence of mRNA. Got it? That is AUG sequence, start codon. Initiator tRNA, that is anticodon, comes and bind with the start codon, that is AUG, on the ribosome. Initiator tRNA आता है और mRNA के start codon AUG पर attach हो जाता है. So mRNA has the codon that is in the bases which are complementary. So tRNA carry complementary bases present on the anticodon, right? तो tRNA पे जो है anticodon present होगा, जैसे UAC, right? So in initiation, codon of mRNA bind amino acid codon of tRNA, starting with the AUG, and then start the process of translation begins. Now coming to the next step, that is elongation. What happens during this stage? One by one tRNA come and attach with mRNA, okay, according to the complementarity, and keep on adding amino acids one by one and form the growing polypeptide chain, right? Now coming to the next stage, the final stage, that is known as termination. When tRNA comes to the stop codon which is present on the mRNA, then the process of translation becomes finished, okay, it becomes terminated. So termination is when tRNA comes in the codon of 3 stop codon. tRNA के कोडोन्स जो है 3 स्टॉप कोडोन, याद है आपको, मैंने अब 3 आपको स्टॉप कोडोन बताए थे, UAA, UGA और UAG. जब इनके कोडोन इनके कांटेक्ट में आता है तो ये प्रोसेस ट्रांसलेशन की वहीं रुक जाती है. सो टर्मिनेशन इज बेसिकली द प्रोसेस ऑफ स्टॉपिंग द ट्रांसलेशन. ओके, नाउ व्हाट इज अ पेप्टाइड बॉन्ड? Peptide bond is a chemical bond formed between amino group and carboxyl group. I think we have discussed, right? We have discussed in class 11th also, we have discussed earlier also. Then finally the formation of peptide bond is there, and this formation of peptide bonds required energy which is provided in the form of ATP. Now the very important question, what is polypeptide? Children, polypeptide is the polymer of amino acid. So when many amino acids are joined together by the peptide bond, it is called polypeptide, or in the very simple word, it is proteins. Now coming to translation unit. Translation unit की अगर हम बात करते हैं तो translation unit, it is present in the mRNA, like initiation codon, then stop codon. One we have is initiation codon AUG, then we have 3 stop codon UAA, UAG, UGA, and in between there are many codons, and another is the untranslated codon region. So short form for the untranslated codon region is the UTR, right? So present prior and after initiation codon and stop codons, these base sequences are not translated during protein synthesis. So these UT are present before the 3' end and at the 5' end, right? So before initiation codon and after the stop codon. Now one more point I added along with them, that release factor. What is release factor? Children, during termination, one release factor attach with the stop codon of mRNA and translation ends after that, right? So complete polypeptide chain is released from the ribosome as well as mRNA is also released, right? So that is known as released factor. So when it comes in the contact with the release factor, the polypeptide chain separates, right? So this is about the translation. In the next video, we will discuss about regulation of gene expression. So I hope you understand all the concepts about the translation and genetic code. But still feel any kind of query, so please feel free to ask. So till then, have a nice day and